Hey guys, it's Kenna here. Today we're going to go ahead and continue our discussion of mutations. I know we introduced one last class when we were going over meiosis, and I know we discussed them before when we introduced them two classes ago. But today we're going to go ahead and look at mutations on a bigger sense of scale, talking about the different types, their sizes, their impact, and kind of look at the molecular basis of genetic variation. Okay. So let's start out with our essential questions. Number one, what is an allele? I know we introduced it when we went over mutations before, but let's make sure we know what an allele is. Number two, how are phenotype and genotype related? This question will come up again when we get into genetics, as we'll keep hitting on this phenotype genotype relationship a lot. Number three, what is the difference between somatic and germline mutations? So this is depending on what kind of cells it's in. And we'll talk more about this later, but it's going to tie back into what we talked about last class in terms of meiosis as well. Number four, what are the major mutations by size? Okay. What are the major mutations by effect? Number six, how are chromosomes and chromosome number mutations different from gene mutations? Think about that in terms of their impact. And then number seven, what is a non-disjunction and how does that relate to chromosome number mutations? All right, so let's go ahead and start by this discussing what an allele is. As we mentioned before, an allele is a version of a type of gene. Now, genes occupying the same position on a homologous chromosome are called alleles, and that position is known as the locus. So if you look at our example here, when we go ahead and take a look at chromosomes. So let's say this is the maternal chromosome from mom. This is the paternal chromosome from dad. This is the location for gene A. On the maternal side, we have a dominant A, and on the paternal side, we have a recessive A. Now, any one individual can have a maximum of only two alleles or two versions of the gene because they get one gene from their parent of origin. So, maternal chromosome can only have one gene A, paternal chromosome can have one gene A. They can be different versions, but you only get one from each parent. Okay. This will come into play again when we go ahead and get into genetics and start looking at Punnett squares. All right. So this leads us on to our discussion of genotype and phenotype. Now, I know that these are new words for you now, but you should get used to these because remember, this is going to be a huge player when we get into genetics in particular, but it will also come up when we're going into evolution. Now, if you remember when we talked before about a gene, this came up last term, and I want to just remind you of it. A gene is a sequence of bases in DNA that codes for a specific protein. I think that's a really good definition because it relates genotype, which is the first thing we'll talk about here, to phenotype. Okay, so the genotype is the organism's sequence of nucleotides in DNA. And if you look at this box up here, what we're looking at right here is the DNA. This sequence represents the genotype. The genes are the genotype. And when we go ahead and start moving on, we go through transcription, we go through translation, and when we do that, we end up producing a protein. The phenotype is the manifestation or appearance of a characteristic arising from the individual's genotype. So remember, the instructions are all laid out inside the DNA, but the DNA doesn't do a whole lot other than act as the instructions. The work is carried out by proteins. Your physical traits primarily come from proteins. And we have to go through this process where we take the DNA, transcribe it into RNA, and then convert that into proteins at the ribosome. Okay, so DNA sequence, genotype, protein sequence, phenotype. Okay. And the phenotype is going to vary a little bit depending on the environment and the genetic background of the individual who's being analyzed. All right. Which leads us to mutations. Now, we've been talking a lot about how we really want you to view mutations as a process, all right, not a collection of things. But what's happening is they're looking at changes in the DNA sequence and how they affect that genetic information's ability to convert into a protein that does jobs and impacts characteristics. There are many different types of mutations. Some are more or less impactful than others. The three main types of mutation by size are: one, gene mutations. These only impact one gene. Two, chromosomal mutations. These impact more than one gene on a chromosome. And three, chromosome number mutations. These add an entire extra chromosome or lose an extra chromosome. All right, so what is it that actually causes these mutations to occur? Well, there are two ways in which the DNA can actually become mutated. Number one, you can inherit a mutation. So it came from your parents. This is going to be a mutation we'll talk about a little later that occurred during meiosis. Or two, you can acquire a mutation over the course of your life. These typically come from environmental damage, being around radiation or carcinogens, things like smoking, or a mistake that gets made when DNA is doing replication in mitosis. So, this is a key thing to keep in mind. These cell division processes of mitosis and meiosis end up being prime opportunities for a mutation to occur because we're making that copy, whether it's into the RNA, whether it's into the DNA. And as we do such, we go through this process. It has to copy everyone exactly, otherwise, a change occurs. And if it makes a mistake, that's your mutation. Now, is that a problem for you? Is that a problem for your offspring? Well, it depends on what type of cells the mutation occurs in. Mutations can occur in any cell, and they only affect future generations if they occur in those cells that produce the gametes. Remember the sperm and egg, right? If those mutations go into the sperm and the egg, they can be passed on to future generations. Now, these are what are known as germinal or germline mutations, and germline is the term I'm going to primarily use. Okay. So if you get a mutation, a somatic or body mutation, this is typically something that's going to go to lead to something like cancer, but it stops with the individual. It does not get passed on to the next generation. You can pass on the risk for it, but not the actual problem, the mutation itself. That's very different when we start looking at germline mutations. Germline mutations are those that occur in the sex producing cells. All right. So those cells that produce the sperm, the cells that produce the egg. Now, these mutations, because they are in the sperm, they are in the egg, can get passed on to future generations. And you can end up with individuals who carry only whatever the mutation was that was acquired in those germline cells. Now, I have to say, mutations are always thought of as this dark, deep, horrible thing. But mutations in your body cells in particular are rarely noticed, except in the cases of cancer where they mutate uncontrollably. And we've talked a little bit about the impact with respect to cancer. Mutations in cells other than germline cells are known as somatic or body mutations. And the image that you're seeing here is a color augmented picture of breast cancer. All right. These are cells that are beginning to proliferate outside of their normal tissue of origin, a process known as metastasis that we discussed before. And when we go ahead and look at this, it can become infiltrative, start causing increased blood flow, and cause problems in the development of the tissues around it. Now, mutations are typically going to occur when a copying process is occurring. And for those that are going to be passed on, this process is meiosis. Meiosis is a prime time for mutations to occur. The germline mutations that occur during meiosis are the ones that are going to be passed on during fertilization. Okay, because we're doing these replications so quickly in many cases, there is a chance for a mutation. And while your body has fact-checking enzymes that look for mistakes, they don't always catch them. So what type of mutations are we looking at in terms of size? Well, as we mentioned a little while ago, these are a gene mutation, which is changes to the bases of a DNA in just one gene. We can get a chromosomal mutation, which affects the whole or large part of the chromosome. And we can get a chromosome number mutation, which has additional or fewer chromosomes than are traditionally present. So let's begin by taking a look at gene mutations. In many cases, these are the results of just a single nucleotide being altered, okay? What is typically known as a point mutation. Many gene mutations only impact one amino acid in a protein. Remember, process genotype to phenotype. The mutation occurs in the DNA and gets carried on into the amino acids of the protein, impacting the phenotype. Now, when we have these types of point mutations, they have three different types of impact that they can have on the organism. Okay? These mutations can be what's known as a missense mutation. These produce a new amino acid type. They can be a nonsense mutation, which produces a stop codon, or they can be a neutral or silent mutation, which produces absolutely no change to the actual amino acid sequence. Now, chances are most of you have inside of you some form of silent or neutral mutation, and you don't even know it because it impacts absolutely nothing in terms of the normal functioning of your body. This is not true in terms of the long-term impact of evolution and the accumulation of new variations and this type of thing. Hopefully, you also recognize that a nonsense mutation of all of these is definitely the most impactful. When we were going ahead and using our codon wheel to help go through transcription and translation, when you hit a stop codon, what happens? Hopefully, you said that it terminates the sequence. There is no amino acid, it ends it. Now, if you have a really long amino acid chain for your protein, and you all of a sudden have a mutation that causes a stop codon halfway through, your protein is now half as long. And what is the likelihood that that's going to actually be effective in performing the function of that particular protein? Yeah, probably not that good, which is why when we start looking at things like lactose intolerance is a good example. A mutation occurs, changes the protein structure, either at a key folding point in a missense substitution or in a nonsense mutation that's going to go ahead and produce a stop codon, and you're missing half the protein. Now it's not going to be able to do his job as well or even at all. And then you have the impact of something like lactose intolerance. Okay, so let's start by taking a look at like a neutral mutation. Some mutations don't really have an impact on the organism. They're not beneficial, they're not harmful, they just occur. And these are known as neutral or silent mutations. If you go ahead and look at our example on the right, we have an amino acid sequence of phenylalanine, tyrosine, glutamic acid, and valine. And then you look on the bottom, and the amino acid sequence is exactly the same. This is because when we start looking at our triplets, remember there is some generic nature in our codons that means that multiple codons code for the same amino acid. This mutation changed to the same amino acid, so not to a new one, and your body doesn't even know the difference. Now, this type of mutation may have no immediate adaptive value when it occurs. It's not helpful, it's not hurtful, but this may not be the case in the future. Neutral mutations are really hard to detect because they don't produce a change in the phenotype. So when we go ahead and take a look at this example, you see in that third triplet, CTT, the mutation occurs that replaces that second T with another C. So now we have CTC. This changes the mRNA from GAA to GAG, which you see, if you look at your codon wheel, produces the same amino acid. So, what kind of mutations are most likely to occur? Well, we typically start looking at mutations in single bases in terms of, is it a purine going to a purine? Is it a pyrimidine going to a pyrimidine? Or is it a purine to a pyrimidine or a pyrimidine to a purine? If you do what's known as a transition, the body is less likely to catch it because the spacing in terms of the bases may not be largely different. A purine double ring base replaced by another double ring base, less likely to get caught. A pyrimidine being replaced by a pyrimidine, less likely to be caught. But it's more often seen that we say have transversions, which convert a purine to a pyrimidine, and then the body typically is going to go ahead and catch those. That's not always true, but because there is a difference in terms of the actual spacing of the rungs of the ladder of the double helix, your body is more likely to go ahead and catch these types of mutations. All right. But some mutations are actually bigger than this. So it's not a point mutation. It's going to impact more than just one gene. And for these bigger genes, bigger mutations, we call them block mutations. When we say block, what we mean is it impacts more than just one gene on a chromosome. Now, what kind of mutations are we talking about? Well, you can still have an insertion, but we call it a duplication because it's typically more than just a gene's worth. We can have a deletion. Okay. These kind of changes can have a big impact because it gets rid of whole genes. If you take a look at the example down here at the bottom, you'll notice that it points out that there are some breaks. And this may happen for various reasons. Maybe it's a crossover like we talked about before, or maybe something happens and there just is a damaged piece of chromosome and the body tries to go ahead and cut it out using enzymes. Sometimes when you do these cuts, rather than going ahead and repairing them and putting them back where they're supposed to, sometimes let's say you lose a section. If you lose a section of the chromosome, the genes that it represented are gone. And so the other chromosome is going to have to compensate in terms of gene production for those particular proteins or not be used at all. And these aren't the only types of these chromosomal block mutations. We also have, as I mentioned, your deletion, where you lose a gene, your duplication, where you add an additional gene, a copy of one you already have. You can get an inversion, where the chromosome gets twisted up, it gets cut and put back together incorrectly, and now the genes are reversed in order. This can definitely cause some problems. And then we have translocations, like your crossovers, where you actually are going to exchange genetic material from one chromosome to another. So how do these things kind of happen? Well, the easiest examples to look at include like duplication or deletion. And when we start looking at things like simple base point mutations, this is how a lot of those occur. If we're going to go ahead and synthesize a new strand, we're going ahead and doing DNA replication. Our DNA polymerase is copying from 3' to 5'. Sometimes it gets going through and it ends up copying something twice. It slips and then it bunches up or something along those lines, and you end up with an extra copy of the same nucleotide. Now you've got an extra one which changes all the rest of the triplets and all the triplets after that. Likewise, sometimes the strand that's being copied will slip or get bunched up, and you will end up making a copy that is now missing a particular nucleotide base. And that changes the triplets from there on out as well. So strand slippage can definitely have a big impact on the outcome of a particular sequence and hence its protein. The last type of chromosome, the last type of mutation by size I wanted to highlight is that of the chromosome number mutation. These are also known as what are called non-disjunction mutations because they don't do the normal separation process that occurs when we go ahead and take a look at anaphase. And this can lead to one of two things, either aneuploidy, where it's the loss or gain of a whole chromosome, good example of which you see below. Typically, most individuals have 2 copies, one from mom and one from dad for chromosome 21. But from time to time, you'll end up with an individual who has 3 copies. This is known as trisomy 21 or Down syndrome. Another example of this is polyploidy, where you don't just lose or gain one chromosome, but you lose or gain a complete set of chromosomes. Now, this is not something that we see in animals because this is typically going to go ahead and end the life of the organism. But this is something that we do see in plants. If you have ever had heirloom roses, the really giant roses that exist that a lot of people have in their yards, these are a good example of polyploidy. They actually double the number of chromosomes inside these organisms. And what they found is that it actually produces organisms that have larger blooms, that are more fragrant, that are more colorful. Now, that's a beneficial adaptation almost when we start looking at plants, but it kills animals. You might take a minute and try and think about why this could be. Now, when you get these abnormal numbers of chromosomes, okay, I'm not going to talk about polyploidy, I'm going to focus more on aneuploidy. We are ending up with something other than the normal number. Now, typically we would see two, one from mom and one from dad, and this is known as disomy. But aneuploidy is a condition where we're missing or adding extra chromosomes to those numbers. Now, when we go ahead and see this, there are tons of examples that we can go ahead and highlight. The first of these is what's known as a nullisomy. This is where the individual gets none of a particular chromosome. They didn't get one from mom, they didn't get from dad, they just don't have any copies. So they can't get any of the genes or the proteins those genes produce. More commonly, you would see something known as monosomy, where they only have one copy. They get it from mom or they get it from dad, but they don't get two copies. And whatever is on that one copy from the parent they get it, that's what they get. I'm going to skip disomy because disomy is what most people have. That's the normal condition. And then skip on to trisomy. We gave you the example of Down syndrome, trisomy 21, but there are many different trisomies. And when we get on and start looking at genetic engineering, we'll show you some examples of these. But we can start looking at sexual trisomies when we start looking at things like Klinefelter's disease, or we start looking at metafemale. Or you can go ahead and look at examples of monosomy, things like Turner's syndrome fall into this category. And I'll go ahead and provide you a link so that you can learn a little bit more about some of these examples. The next one is tetrasomy. Now, this is a little less common than the others. It's more like nullisomy. It does happen, but it's not frequent. And this is where you get 4 copies. So you got 2 copies from each parent, giving you double the number of copies you got for a particular chromosome. And the vast majority of these aneuploidies result from a non-disjunction during meiosis. So let's give you some examples of non-disjunction. Now, what happens, if you remember your notes from last class, in meiosis is that the spindle is going to separate the homologous pairs in anaphase 1, or it's going to separate the chromatids in anaphase 2. However, sometimes these processes don't occur as you would expect them to. And when these types of mistakes happen, the failure to separate during anaphase produces this extra or missing chromosome. Okay? This mishap is known as a non-disjunction and can occur in either meiosis 1 or in meiosis 2. So on the right here, what you can see are 3 different examples of trisomies: trisomy 13, trisomy 18, and trisomy 21. Now, we've already talked about trisomy 21 because that's the one that causes Down syndrome. In trisomy 13, trisomy 18, these are typically inherited diseases that can be problematic in development. And in both cases, the individuals born with these typically do not survive past the age of 2 or 3. When we go ahead and look at this, my older son was born with a nevus, and that's a result of what we identified in our ultrasound called a choroid plexus cyst. Now, when they saw that, they were worried that my son was going to have one of these trisomies. And so we had a lot of genetic counseling but decided to not go ahead and get tested in the long run because it wasn't going to impact our decision to keep the baby. I'm happy to say that my son is about to turn 10 and he's doing just fine. And so this was not a problem that we ended up dealing with. All right. But these types of non-disjunction results are more common as maternal age tends to increase. Um, and you can end up passing these on in either the sperm or the egg, depending on what we end up seeing. If either of these abnormal gametes ends up uniting in the process of fertilization that we talked about last class, the offspring is going to have an abnormal number of chromosomes, which leads to an aneuploidy, including trisomies and monosomies. So let's take a look at non-disjunctions, both in meiosis I and meiosis II. We'll start with meiosis I. So a non-disjunction in meiosis I occurs when homologous chromosomes fail to separate during anaphase I. You can see your homologous pair, instead of separating to opposite sides, two of them get stuck together and both go to the same side. Well, what does that lead to? In meiosis II, though, it does a normal separation, but now you'll notice that meiosis II produces all of the cells are going to have an abnormal number. The two on the left have an extra chromosome, and the two on the right are completely missing a chromosome. This is due to a non-disjunction, and any of these gametes, whether they're sperm or egg, will produce an aneuploidy. Well, what if that occurs in meiosis II instead of in meiosis I? Well, the process is similar, but remember that we're not separating homologous chromosomes in this case, but instead we're going to separate out chromatids since we're in meiosis II. So meiosis I goes along normally and nothing happens incorrectly. But when we get into meiosis II, the point where we would normally separate out the chromatids doesn't occur properly. But in this particular case, instead of producing 4 gametes that are going to produce abnormal aneuploidies, we end up with only 2 out of the 4. Okay, one that has too many, one that has too few, and 2 that are normal in terms of their number. Now, I'd like you to pause the video at this point and think a little bit about how this is going to impact production in a male and in a female. Does this matter when we're going to go ahead and start looking at production of gametes? Think about what a polar body is and spermatogenesis and oogenesis. Okay, so take a minute, think about that, and then we'll finish up. Okay, so hopefully you start to realize that there is a little bit more risk here when we go ahead and start looking at this particular example for females to have less problems because they only produce 1 egg. And now it could be 25% of being any one of these. And so we start thinking about that. So maybe we're more lucky. It's hard to say, but I want you to think about the impacts. Let's finish off by taking a look at fitness and mutation rates. Now, genes typically mutate at known rates, and this rate varies depending on the size of the gene. The bigger the gene, the greater the risk for mutation. And the location of the gene and the sequence of the gene will also impact its risk of mutation. Some genes have very high spontaneous mutation rates because of their sequence or location. With approximately 30,000 genes in the human genome and 2 copies of every gene, then an average of 1 in 5 of us carries a brand new mutant gene, one that's never been seen anywhere else in the entire world. But remember that when we start saying a new mutant gene, that a lot of those don't have an actual impact on the outcome. Okay, not a large impact at least. Mutation rates for genes within a species are probably similar, but the viability of these mutations can vary greatly. This is where we start talking about fitness, which again is going to be a hugely important term when we go ahead and take a look at evolution. The fitness of a mutation describes its value in terms of the survival and reproductive success of a particular organism. A mutation may turn out to be lethal, meaning that the individual isn't even born in many cases. These are non-viable. It can be harmful, which it's not a lethal mutation, but it can cause some serious impacts to the organism and lower their chance of survival and reproduction. So, good examples of this include things like Down syndrome or sickle cell anemia. Then we have a whole bunch of mutations that are identified as silent. Most of these point mutations are absolutely harmless because they have no effect on the overall phenotype. And then we have the beneficial mutations. Now, occasionally, one of these mutations will actually be useful or very useful in a particular environment. Uh, Examples include insecticide resistance in insects, antibiotic resistance in bacteria. These mutations then give an advantage to the individual who has obtained it and get passed on to future generations as something that's helpful. Okay, so a couple key takeaways from today. One, all new alleles or all new variations of a gene originate by mutation. Two, new alleles introduce genetic variation into a species, which can change their ability to survive in a particular environment. This is something that natural selection can act upon. We'll talk about more about natural selection when we go ahead and get into evolution. Most mutations that occur are in somatic cells or body cells, and therefore are not inherited. We give the example of cancer. Okay. There are, however, mutations that are passed on to offspring. These are mutations in the gametes, which we call germline mutations. These can be passed on to future generations, and their impact depends on its size. Okay. Is it a chromosome mutation? Is it a gene mutation? Or is it a chromosome number mutation? Or based on its impact, is it a missense mutation? Is it a nonsense mutation? Or is it a silent mutation? All right, guys, I hope this has been helpful and you've got a little introduction to the major topic of mutations. This will come up again from time to time in our genetics unit when we get to it in a little later in this term. All right, guys, have a great rest of your day. Take care of yourselves, stay safe, and I'll talk to you soon. Bye bye.